Have you ever felt like this bear relaxing on the river? Well, there are significant issues you could be dealing with if there's Shigella in the water. Shigella is a gram-negative, non-spore-forming, non-motile, bacilli-shaped bacteria. Shigella is a chemoorganotroph and a facultative anaerobe and is often cultured on mac agar, which helps differentiate it from others based on its inability to ferment lactose. This characteristic is often used in lab settings to help differentiate Shigella from other bacteria such as E. coli, which can ferment lactose. Numerous gram-negative bacterial pathogens such as Shigella and Salmonella employ the T3SS mechanism to infect hosts. Upon contact with the host, the T3SS inserts its translocon pore into the host membrane, creating a channel for the delivery of effector proteins into the cell. You can see that here on this diagram. Transmission of Shigella often happens with dirty water, hand-to-mouth situations, and often in food handling. These bacteria have rapidly acquired resistance to multiple antibiotics through horizontal gene transfer, including quinolone resistance. Addressing Shigella infections requires a multifaceted approach, including improving water sanitation, monitoring antibiotic resistance, and enhancing public health infrastructure to prevent outbreaks. Let's try to keep our water clean so we, like this bear, can enjoy all that nature has to offer.
Tuberculosis Tales, Mycobacterium's Whispered Trails. I am the cause of tuberculosis, TB, which the World Health Organization, WHO, classified as a deadly respiratory disease after I threatened the world in 1993. In fact, I'm so famous that the WHO publishes epidemic reports all about me every year. The 2019 report estimated that 10 million people were infected and around 1.4 million people suffered mortality. According to some models, the COVID 19 epidemic may worsen TB burden due to undernutrition, multi drug resistant TB strains, unemployment, and redirection of resources to fight COVID 19. Who am I? Mycobacterium tuberculosis. I am a gram-positive, acid-fast bacillus with a very tough cell wall composed of lipopolysaccharide, mycolic acids, arabinogalactan, a polymer of arabinose and galactose, a thick peptidoglycan layer, and a lipid bilayer. Over 60% of my cell wall is made of mycolic acids, which contribute to my virulence. Other lipids in my cell wall, such as sulfolipids, cholesterol, and liporabinomannine, also help to increase my virulence. They help me be resistant to antibiotics, resistant to osmotic lysis, and oxidative stress. I hail from the bacteria kingdom, Actinobacteria phylum and class, Actinomycetales order, Mycobacteriaceae family, Mycobacterium genus, and tuberculosis species. I have several other relatives that form what is called the tuberculosis complex, because we all cause tuberculosis disease in either humans or animals. They are M. africanum, M. bovis, M. mungi, M. canneti, M. origus, M. microti, M. caprae, and M. pinnipedi. One of my relatives causes leprosy, and their name is Mycobacterium lepra. Not all of us cause tuberculosis, though, such as Mycobacterium smegmatis and Mycobacterium fortuitum, but they can cause human infections. Humans are my only known host, and I am spread primarily through respiratory droplets, such as sneezing or coughing. I am famous for invading the alveolar sacs of the lungs, but also invade the lymph nodes, bones, joints, central nervous system, heart pericardium, or bloodstream. I cause humans to cough, have chest pain, lose weight, fever, and even night sweats. If in the lymph nodes, the person can get abscesses. If in the central nervous system, I get quite a headache. My metabolism is heavily dependent on lipids, which are my primary carbon and energy sources. I'm such a chemoheterotroph. I steal lipids from my host because they are delicious, but I can also make my own. In fact, I can use cholesterol as my sole carbon source. Important enzymes responsible for fatty acid biosynthesis are FAS1 and FAS2, which produce some mycolic acids that make up part of my cell wall, such as PDIM, sulfolipids, polyacyl trehalose, and diacyl trehalose. Scientists predict that under aerobic and early growth conditions, I use glucose and triglycerides as carbon and energy sources, but then switch to using lipids once inside host macrophages where I cannot access glucose. Take that, macrophages, I can still replicate. Sometimes, a bunch of macrophages will chase me down and form granulomas by clustering together in large groups of foamy cells which starve me of oxygen. I must change up my metabolism if I want to survive, so I alter my Krebs cycle to favor succinate production. Succinate feeds succinate dehydrogenase, which is involved in both the Krebs cycle and electron transport chain. This means I still get to make ATP. To increase the success of my invasion, I must reduce the population of my enemies. Studies in TB patients have shown that I am associated with a decrease in microbiome diversity, especially short-chain fatty acid or SCFA-producing bacteria such as firmicutes and Bacteroides. You see, SCFAs, such as butyrate, are helpful in immunomodulation, meaning that they help stimulate lymphocytes and phagocytes. I don't want that. Gut and lung dysbioses are my friends. I don't dislike all microbes in the human microbiome, though. I do like proteobacteria, so they are overrepresented during MTB infection. During anaerobic growth, they produce mixed acids, including succinate, lactate, formate, but also dreaded SCFAs. However, as I mentioned before, I do like succinate for my own ATP production. I also like some actinobacteria, such as from the genus Collinsella. No one knows exactly why these groups are overrepresented, and I'm not telling. I have many clever ways to escape the human immune system. One way is by purposely allowing myself to be internalized by alveolar macrophages, but then preventing the maturation of the phagosome. You see, during normal phagocytosis, the phagosome fuses with the lysosome to form a phagolysosome, which contains an acidic environment and enzymes that can kill me. One way I prevent lysosomes from killing me is by secreting a lysosomotropic, can penetrate lysosomes lipid called 1-TBAD, which functions as an antacid. Another way I do it is by secreting a protein, tyrosine phosphatase, PTPA, which prevents the ATPase in macrophages from doing its job of actively pumping protons and acidifying the phagosomal environment. Since I can damage phagosomes with my ESX1 effector proteins, macrophages try to come and repair them via the endosomal sorting complex required for transport, or ESCRT, and Gal3 proteins. However, my handy effectors ESXH and ESXG interfere with the recruitment of ESCRT. This prevents maturation into a phagolysosome and thus keeps the pH high enough to keep me comfy. Humans have found that ESXH and ESXG complex with each other and prevent ESCRT recruitment, thus disrupting its function, which also includes moving me to lysosomes. Gal3 gets recruited to damage phagosomes when internal glycoproteins get exposed, but my effectors block that signaling too.
My name is Josh, and today I'll be presenting on Bordetella pertussis, the causative agent of whooping cough. Bordetella pertussis has a long history as an infectious agent and is known as the causative agent of the disease whooping cough, otherwise known as pertussis. The first recorded outbreaks were in Paris during the 16th century by Guillaume de Bayeux. However, it wasn't until 1906 that Jules Bordet and Octave Zangu successfully isolated this bacterium. Before the first vaccines were introduced in the 1940s, the US saw over 200,000 cases annually. Post-vaccine cases reduced by more than 75%. However, whooping cough remains a global issue, with 24.1 million cases and 160,700 deaths in children under 5 annually. This is an image of Bordetella pertussis growing on the medium created by Bordet and Zangu to isolate the first Bordetella pertussis bacteria. The medium includes glycerin extract of potato, agar, salt solution, and rabbit or human blood. Bordetella pertussis is a small, gram-negative, strict arrow coccobacillus, approximately 0.8 micrometers by 0.4 micrometers. It's non-spore forming and typically encapsulated, although it can form a slime sheath layer instead of a capsule, typically in vitro or in a lab setting. A unique feature is its lipo-oligosaccharide composition. The lipo-oligosaccharide of Bordetella pertussis includes a pentaacylated lipid A linked to a complex core trisaccharide. This structure, while potent as an endotoxin, lacks an O antigen, which differentiates it from many other bacteria that have standard lipopolysaccharide composition. This loss of O antigen is thought to be a result of genome decay, or the loss of genes, as Bordetella pertussis has evolved from one of its ancestors in the Bordetella genus. Bordetella pertussis belongs to the genus Bordetella and the family Alcaligenaceae. It's part of a group of prominent Bordetella species referred to as the classical Bordetella species, including Bordetella pertussis, Bordetella parapertussis, and Bordetella bronchoseptica. Phylogenetic analysis suggests that Bordetella pertussis evolved from Bordetella bronchoseptica, which thrived in soil and water environments using amoeba as hosts. Unlike Bordetella bronchoseptica, Bordetella pertussis exclusively infects humans, which is again likely due to genome decay eliminating its environmental survival capabilities. Instead, Bordetella pertussis retains a unique ability to survive within macrophages. Despite genome variations, classical Bordetella species share several virulence factors. Adhesins like filamentous hemagglutinin, pertactin, fimbriae help the bacteria attach to ciliated epithelial cells. Toxins such as pertussis toxin and adenylate cyclase toxin disrupt the host immune response by destroying ciliated cells and inhibiting phagocytosis. Inhalation of airborne droplets containing Bordetella pertussis starts the infection. Bacteria attach to ciliated epithelial cells in the nasopharynx and trachea using FHA, protectin, and fumbriae. They then replicate and produce toxins. Tracheal cytotoxin destroys ciliated cells, inhibiting the ability of the host to remove mucus properly, causing that distinctive and characteristic whooping cough. PT inhibits immune defenses, and ACT disrupts cellular signaling, inhibiting phagocytosis and inducing apoptosis of macrophages. This is a figure I drew that illustrates the transmission steps I just described of Bordetella pertussis, from inhalation to toxin production and immune system interference. Bordetella pertussis has unique growth requirements and metabolic characteristics that enable its survival in the human respiratory tract. It thrives in aerobic conditions with a temperature range of 35 to 37 degrees Celsius and a pH range of 7.0 to 7.5. Nicotinamide supplementation is essential for its growth. However, the presence of fatty acids, metal ions, sulfides, and peroxides can inhibit its growth. Unlike many bacteria, Bordetella pertussis cannot use carbohydrates as its primary energy source due to missing genes for functional glycolysis. Instead, it relies on gluconeogenesis to produce glucose, which is vital for creating the lipooligosaccharides in its outer membrane. The primary carbon sources for Bordetella pertussis are amino acids, with glutamate being the most efficiently metabolized. This adaptation allows it to fully utilize the TCA cycle, an electron transport chain for energy production, using oxygen as the final electron acceptor. Understanding these metabolic adaptations helps explain how Bordetella pertussis thrives in its specific environment and provides insights into potential targets for therapeutic interventions. Bordetella pertussis infections are typically treated with erythromycin, which blocks protein synthesis, reducing bacterial growth. Interestingly, the host microbiota also plays a crucial role in defense against infection. Studies have shown that removing nasal microbes allows efficient colonization by Bordetella pertussis, while antibiotic-induced microbiota dysbiosis increases bacterial burden and impairs immune responses. In conclusion, Bordetella pertussis has a significant historical and contemporary impact as a pathogen. Its evolution from an environmental microorganism to a human-specific pathogen showcases its adaptive capabilities. Understanding its interactions with the host and the microbiota provides insights into disease dynamics and potential therapeutic strategies. Ongoing research is essential for developing more effective prevention and treatment methods across the globe. Thank you for your attention. For further reading, please refer to the listed resources.
E. coli is a bacteria that commonly lives in human and animal intestine. It is a gram-negative, rod-shaped, and facultative anaerobic bacteria, and this organism was first described by Theodore Skerek in 1885. And when we say it is facultative anaerobes, it means that it grows in the presence and absence of oxygen. So it prefers the presence of oxygen, but in the absence of oxygen, it is still these microorganisms can survive. Most of the E. coli species is harmless. Escherichia coli is a non-competent bacteria, meaning that it doesn't naturally take DNA molecules from the environment. In order to take the DNA molecule from the environment, it must induce into artificial competency. This bacteria has flagella, fimbriae, and a pili. This pili is used for transferring the genetic material from one E. coli to another. It requires nutrition and energy for survival. Some source of that energy is from the carbon source, from the phosphorus and sulfur source. In terms of energy, the E. coli gets energy via glycolysis process, and it interacts with other microbes in its surrounding, includes soil, water, animal gastrointestinal tract, and many diverse environmental needs. This interaction could be cooperative or competitive in the ecosystem. When we say it is competitive interaction, it means that it competes for food, space, and resources in the environment with other microorganisms. So, cooperative interaction means the mutual relationship that E. coli has with the other microbes in the ecosystem. Generally, the E. coli is commonly found both in human and animal intestine, while most strains of E. coli are safe and play an important role in digestion. Few can cause disease ranging from minor to severe infection. Many research has been done regarding E. coli, such as the research that Laurent Porel and his fellow conducted research on antimicrobial resistance in E. coli. They summarized that resistance to multiple drugs in the E. coli has emerged and has a major concern. Another study on competition among E. coli strains for space and resource indicate that E. coli faces intense competition in environment due to the scarcity of food, space, and other resources. So many other study has been done regarding E. coli due.
Hi, I'm Tara, and this is my term project on Lactobacillus acidophilus. You can see my works cited page here, and you'll notice each source corresponds with a Roman numeral, which will pop up throughout the comic. I wanted to approach this with a really general audience in mind, doing my best to keep all the information as accessible as possible. Here we go. Hello? In here. Closer. Hi, I'm Lactobacillus acidophilus. Sorry it took so long to find me. I'm pretty small, like only 2 micrometers small. Anyway, a bit about me. I'm rod-shaped, gram-positive, low GC, catalase negative, and non-sporulating. I pretty much look like a Cheeto Puff under the microscope, and I'm often arranged in chains or palisades. You won't be able to see me here, but I'm in a lot of foods and supplements. More about that later. For now, let's slow it down and start from the beginning. I was initially isolated from infant feces in 1900. I know, but we all start somewhere. That said, I've existed long before humans isolated me. In fact, I've probably been around since before humans were a thing. Everyone in my family is part of the domain Bacteria. My great-great-great-grandmother belongs to the order Lactobacilliales and hangs out in the same acid-tolerant circles as Streptococcus and Enterococcus. My closest family belongs to the order of Lactobacillus. I have like 170 cousins, and we all kind of look alike, so it can be hard to tell us apart just by looking. I grew up indigenous to the human gastrointestinal tract, but I have a ton of family in the reproductive tract. L. iners, L. crispatus, L. jensenii, and L. vaginalis have lived there since before I can remember. And I've been to visit family up north in the mouth as well. Most of my family has a reputation for being friendly and helpful to the human body, but there are the occasional opportunistic pathogens. Even I can cause issues from time to time. Dental abscesses, endocarditis, empyema, and sometimes even infections in prosthetic limbs. Sorry. All that aside, I'm an overwhelmingly friendly microbe, and that's what we'll focus on today. I'm mostly known around these parts for my ability to maintain acidic environments and restore microbial balance. How do I do it? Well, to start, I love carbs. Who doesn't? I especially love lactose, and I'm really good at fermenting it into lactic acid. If I'm being honest, carbohydrates are the only energy source I can eat, and lactic acid is the only thing I can produce. I'm what they call a homo fermenter. It's why I live where I live, in acidic zones, typically ranging from 5.5 to 6. Push it past 4 on the pH scale, though, and I'm out. I prefer heat and moisture. 37 to 42 degrees Celsius is my favorite. Out of all of my closest relatives, I'm the least tolerant of oxygen, so most people call me microaerobic. But I really prefer anaerobic environments. As far away from oxygen as possible, please. All this to say, I've got what's known as desirable probiotic physiology, which is really just a fancy way of saying the way I'm built makes me a beloved bacillus in the world of probiotics. I'm good at what I do. My bread and butter is maintaining those acidic environments, which defends my host from harmful pathogens who can't stand the acid I thrive in. I'm also bile resistant. There are also a lot of studies that say I'm good at mediating immune response, lowering serum cholesterol, improving lactose metabolism, and treating or preventing infection. People turn me into freeze-dried granules, powders, capsules, liquid preparations, and even vaginal suppositories. Still not convinced I'm the GOAT? Check it out. I regulate the balance of intestinal flora by reducing intestinal pH and producing metabolites. I give pathogens like these guys a really hard time. I can also inhibit growth of pathogenic microorganisms that produce harmful exoenzymes, like azoreductase, nitroreductase, and beta-glucosidase. I can even simply reduce enzyme activity altogether. I also love to adhere to colonocytes, making it tough competition for pathogenic bacteria. And just to re-emphasize, I'm really helpful within the reproductive tract of estrogenic people. I'm really efficient at restoring balance and addressing issues related to bacterial vaginosis. Really, really. Want to stay in touch? Me too. You can find me in milk and yogurt enriched with me, in miso, tempeh, and fermented foods. Make sure to eat things like onions, garlic, bananas, honey, barley, and wheat. They contain prebiotics I love. Bye!
Hi, this is Iris from microbiology. I chose Rhizobium as my favorite microbe. Nitrogen makes up around 78% of all atmospheric gases. Almost all of that nitrogen exists in its diatomic form, N2. No plants or animals are able to utilize nitrogen directly. The only organism capable of utilizing nitrogen are groups of prokaryotes called diazotrophs. Some plants have evolved to form symbiotic relationships with diazotrophs. The main example of this is legume rhizobium symbiosis. Rhizobia get nutrients from the cells of legume roots and fix atmospheric nitrogen into ammonia, which is used by their plant hosts. Rhizobia that exist in free living forms in the soil are incapable of nitrogen fixation. However, during symbiosis, rhizobium undergoes physiological and structural changes that allow it to do so. Bacteria in the genus Rhizobium are rod-shaped. They usually have flagella and are mobile. Once it's in the legume host, Rhizobium transforms into an irregular branch shape. Rhizobia are gram-negative genus of bacteria. Rhizobium's traits of nitrogen fixing and legume association are not phylogenetically significant. Some Archaea, cyanobacteria, and several other bacterial groups who are not phylogenetically related to Rhizobium also have the ability to fix nitrogen, which suggests that this trait is a result of convergent evolution. Rhizobium's capacity to associate with legumes is most likely the result of horizontal gene transfer amongst bacteria, as opposed to vertical gene transfer, which is much more phylogenetically significant. The Rhizobia group has been described as both paraphyletic and polyphyletic. The Rhizobium genus splits into 3 subgenuses: Rhizobium, Bradyrhizobium, and Azorhizobium. All 3 fall under the category of proteobacteria, but on distinct branches, each of which also includes bacterial species that are not Rhizobia. Rhizobia are heterotrophs. Heterotrophs are organisms that cannot make their own food. Heterotrophs get their nutrients from other sources of organic carbon. Rhizobium is a chemoheterotroph, which means it gets energy and nutrients from preformed organic energy sources, which have been synthesized by other organisms. Rhizobia that exist in free living forms in soil feed on dead organic material and use it for growth and to provide substrates that are oxidized in cellular respiration to generate ATP. When Rhizobia form symbiotic relationships with legumes, the photosynthetic hosts provide them with carbohydrates and all the necessary nutrients except nitrogen, which Rhizobium obtain from the atmosphere. Even within individual species of Rhizobium, specific strains can either be mutualistic, parasitic, or non-symbiotic. Mutualistic Rhizobia provide hosts with lots of fixed nitrogen. Parasitic Rhizobia rely on hosts, but fix little to no nitrogen to provide to their hosts. Non-symbiotic Rhizobia only exist in free living forms in soil and are unable to infect legumes at all. Legumes favor more mutualistic Rhizobia. Mutualistic and parasitic Rhizobia can produce millions of descendants. However, the rate of viability among said offspring is very low. All in all, non-symbiotic Rhizobia often have more successful rates of reproduction. So here is an in-depth look step-by-step at the relationship between Rhizobia bacteria and legumes. The process of root formation is initiated when a legume releases a chemical including amino acids, dicarboxylic acids, and low concentrations of excreted components called flavonoids through their roots, which the Rhizobia in the soil are attracted to. Rhizobium accumulate near the root hair and eventually attach to the root hair's cell wall. Chemicals called nodulation factors are released from the Rhizobium cells, which cause the root hair to curl. Now the Rhizobium cells are able to digest the cell wall of the root hair and enter into the root hair. Rhizobium enters the root hair through invagination, which is the process of the cell membrane of the root hair being turned inside out or folded back on itself to form a cavity. The Rhizobium bacteria enter the root through this formed cavity, which is termed the infection thread. Spontaneously, the cells of the pericycle layer of the cortex of the root become dedifferentiated, which leads to the formation of masses of undifferentiated cells called nodule primordia. The nodule primordia divide to produce outgrowths called root nodules. The Rhizobia in the root nodule cells undergo physiological and structural transformation and are now called bacterioids. So the bacterioids are irregular shaped, non-flagellated, branched, and non-dividing forms of Rhizobium. They produce the enzyme nitrogenase. Nitrogenase is responsible for the actual fixation of nitrogen into ammonia. This enzyme is very sensitive to the presence of oxygen and will actually become inactive when exposed to any oxygen. To avoid this, the bacterioids and the legume cells work to form a pink pigment, leg hemoglobin, that is an oxygen scavenger that removes any excess oxygen from the nodule cells. So that's how that works. The symbiotic relationship between legumes and rhizobia fuels the practice of crop rotation, which is used in many agricultural traditions. Crop rotation with legumes allows farmers to nourish plants who can in turn nourish the soil in a process that uses a biological catalyst at room temperature and ordinary pressures and uses sunlight to drive it. Farmers have been using legumes in agriculture since the ancient world to rotate crops to assure the fertility of their soils. In the New World, farmers use the common bean. In the Mediterranean, they use lupins. In China, they use soybean crops. In South Asia and Africa, cowpea was used. Crop rotation with legumes takes the place of relying on industrial nitrogen fertilizers, which is in the next slide. Overuse of nitrogen fertilizers can lead to the release of greenhouse gases like nitrous oxide and the eutrophication of waterways. Nitrous oxide's warming potential is around 300 times greater than that of carbon dioxide. Eutrophication is unwanted fertilization of a waterway that can lead to overgrowth of ecologically harmful microorganisms like algae and cyanobacteria, and eventually lead to the loss of all other aquatic life. The use of legumes that are symbiotically associated with rhizobia, which are naturally occurring soil nitrogen fixing bacteria in crop rotation practices, may help reduce the amount of industrially fixed nitrogen that is used, and that leads to loss of nitrogen into our atmosphere and waterways. So in conclusion, here's a picture of my back garden bed. I started growing some peas recently and have been very pleased to learn about rhizobia, which will hopefully continue to be utilized in the future and on a more broad scale as a sustainable way to optimize agricultural food output and reduce reliance on more environmentally harmful agricultural practices like industrially fixed nitrogen fertilizers. This microbe has a lot of potential. Thank you for listening to my presentation. Here are some references for you. And thanks again.
For this presentation, we're looking at the microorganism Helicobacter pylori. Helicobacter pylori is a gram-negative, non-fermenting, non-sporulating microaerophilic bacteria. It's about 2 to 4 micrometers long with a diameter of about 1/2 to 1 micrometers. It has a helical shape with about 2 to 7 Lophotrichous flagella at the end. That means that all the flagella are used for movement in one direction. It does come in a couple other shapes, but the other shapes are not as useful, just in rods or curved. The spiral shape is the one that's most successful and impactful. Helicobacter pylori is within the bacteria domain, and as you can see on this slide, it shows the phylum, class, order, family, and then the genus and species, which is Helicobacter pylori. This microorganism is a highly successful and prevalent bacteria. I saw a variety of different estimates in terms of the prevalence, but one that I saw is approximately 2/3 of all people in the world are believed to have been infected at some point, with higher prevalence in the developing world. The vast majority of people who have this bacteria are asymptomatic, but those who do have symptoms can develop chronic gastrointestinal symptoms. H. pylori is believed to be linked to approximately 89% of gastric cancers, 5.5% of all cancers worldwide, making it the only bacterium linked to development of any cancer. H. pylori has extensive genetic diversity of strains, which helps it thrive. So within one person, there can be a lot of strains, and between different people. The genus Helicobacter consists of over 20 recognized species, with many awaiting formal recognition. In addition to a lot of diversity within humans, there are related Helicobacter species that have been identified in the stomachs of other wild and domesticated mammals. There are a number of features that allow this bacteria to survive and thrive in stomachs. It's microaerophilic, so within the stomach it only needs a little bit of oxygen, and it contains hydrogenase, which it uses to oxidize hydrogen in the surrounding environment to get its nutritional energy. From that hydrogen, it produces oxidase, catalase, and urease. The urease production is key to its survival because it takes it in, breaks it down, and it creates ammonia and carbon dioxide. That ammonia and the surrounding area increases the pH, making it more basic, less acidic, and creates kind of a window of less acidity right around the organism, which allows it to live. When people have an H. pylori infection, it impacts the entire microbiome in a person's stomach. It can be around 40 to 90% of all the microbiota when it's present. So it reduces the overall diversity. The change in pH impacts the ability of other microorganisms to survive. And then another way it can impact the stomach is that eradication therapies can also further ruin the microbiome. It uses its helical shape and flagella to burrow into the mucus lining of the stomach. So it gets through the mucus and up to the skin layer. Once it's in the epithelial layer, it produces adhesins which allow it to stick there and not get washed away. Once it is up against the stomach wall, it stays there and produces ammonia, which decreases acidity. The ammonia allows it to stay there, but the ammonia also is toxic to the epithelial layer, so it ruins this stomach lining. It creates an immune response, more acids. So basically, it sits there against your stomach wall, and then your stomach acid ruins your stomach lining, creating ulcers. With an H. pylori colonization or infection, people can experience abdominal pain, bad taste in the mouth, nausea, heartburn, belching, and the infection is related to a number of diseases: chronic gastritis, peptic ulcers, gastric adenocarcinoma, gastrointestinal reflux disease, and cancer. H. pylori has been isolated in saliva, gastric mucus, dental plaque, and feces. It's transmissible through oral route or fecal-oral route. Right now, there are no vaccines, so hand hygiene, access to clean water are really important. There are a number of different antibiotics that can kill an H. pylori infection or colonization. Other therapies like proton pump inhibitors, bismuth salt, and a number of different foods are recommended. And if you're having all of the symptoms in the previous slide, it might be worth going to a doctor and getting checked out.
Hi, I'm Neisseria gonorrhoeae. I'm a gram-negative Diplococcus obligate anaerobe bacteria. You've probably heard of me from your sex ed class, friends, or maybe even your partner. I'm an obligate human pathogen, which means I love people. I come from a big family, but only one of my siblings shares the same love for people that I do. Her name is Neisseria meningitis. She's the leading cause of bacterial meningitis. She's a little bit less serious about her love for people than I am, but she does seem to cause a bit more harm to her host than I do too. Also, people seem to find out about her faster than they do me. My other siblings in my genus are similar to me, but have other passions. We all like being in wet environments that are about 98.6 degrees Fahrenheit. We all enjoy being on animals' mucosal surfaces, and we are built to thrive in those environments. Before we get into my wants and loves, let's talk about my needs. I'm an obligate aerobe, which means I need access to oxygen to survive and thrive. For nutrition, I'm a little needy. In a lab, I need a special agar to grow. I do best on blood agar and chocolate agar. Yum. In the most basic terms, I need glucose, glutamine, thiamine, phosphate, iron, and carbon dioxide in order to thrive and grow. Now for my favorite topic, people. They've been obsessed with me for as long as I have been obsessed with them. A bunch of famous people have even talked about me, including Hippocrates, who I think is a pretty big deal. I'm even mentioned in the most printed book in history, the Bible. Each year worldwide, more than 80 million people find out that I am with them. And The WHO even recently named me a superbug due to my obsession with humans and adaptability to their treatments. I usually find a new person to be with through the sexual contact of two people. You could say I love love. Once I find a new host, I bind to their epithelium in their throat, vagina, or rectum. When I first get there, I try to hide my best from the host body. Even though I love them, the immune system of my hosts don't really love me back. Depending on which zone I end up in, I use different techniques to hide from the body. In the cervix, for example, I bind to factor H, which the host body identifies as itself so it doesn't attack me. Once I get into a host, I use a few different proteins in my outer membrane called TDTSs that work to hoard some nutrients away from other organisms in the area, to both suppress the other organisms and pathogens and keep some resources for myself in case I need it later. Since I'm exclusive to humans, I had to learn how to adapt to avoid their rude medical advances and efforts to get rid of me. I use biofilms and horizontal gene transfer using my pili to share information with my peers and learn from their previous experiences with these things called antibiotics. Luckily, every time someone survives one of those series, we can share insider knowledge and become stronger and build resistance to these horrible medications. Despite my efforts, scientists are still trying to figure out how to get rid of me. Once I make my new home in my human host, I'm contagious about a week after my first contact with them. Despite that, my host will still test negative for about a week after I'm contagious. That's still not long enough for me, but luckily it takes a lot longer for most people to find out that I'm even there. In female hosts, I typically fly under the radar. I usually do not cause noticeable symptoms in female hosts for a long time. Most find out about me through random routine screening at a doctor's office. I do cause some symptoms in male hosts, but it's usually just some discharge. Even though I love them so much, I can see why they don't like me. If I go undetected for long enough, I can cause some harm to my hosts. 1 in 5 females who do not receive treatment for me will get pelvic inflammatory disease, which can cause scarring and have long-term fertility effects and requires a long course of medication to cure. Additionally, if I'm in a pregnant person, I can cause harm to the baby. I cause neonatal blindness and scarring and ulcers on the cornea of the infant. Thank you so much for taking the time to learn all about me.
Hi everyone. In today's video, I'm going to be talking about the topic of the Staphylococcus bacteria. There's a lot of information to cover for this PowerPoint, so please feel free to pause the video anytime for you to read the information. Thank you. Bye.
So just a quick overview of what my organism is. So Staphylococcus is a gram-positive bacteria. Appears as a dark purple under the microscope. Can sometimes appear in a short chain, but most of the time they look like grapes and how they are clustered together. The size is about 0.5 to 1 micrometer. It is a non-motile. It is also salt-tolerant bacteria. As you can see, this is a drawing done by me. You can see on the left side of the slide or the drawing that they appear to be gram-positive bacteria because they appear as dark purple. They occasionally form a short chain, and also they can be found inside of a healthy patient's nose. Oftentimes they are also found on human skin. So just a little bit about the shape and anatomy. Like I said before, they're spiral shape, characteristic referred to as koki. Clustering, again, occurs due to the way that the bacteria divides. Unlike other koki that might form chains or pairs, Staphylococcus divides into multiple planes. Like I said, as the anatomy, again, they appear as cluster. They contain cell wall, cell membrane, cytoplasm, surface proteins, and teichoic acid. Now we're gonna talk about pathogeny and relative microbial relatives. Staphylococcus is from the family called Staphylococcus. Um, this genus basically has different types of variant species. The most common two are Staphylococcus aureus and Staphylococcus epididymis. Part 1 of the metabolism of my bacteria includes facultative anaerobes, energy generation. Carbohydrate metabolism includes glycolysis and fermentation, which is what we learned in class. Second part of the metabolism is amino acid and TCA cycle in oxidative phosphorylation. The third part of the metabolism is called biofilm formation. As you can see on the right side, right here, is an illustration of how the biofilm forms. Biofilm is a very important part of the metabolism of this bacteria because it not only nourishes them, but it can also protect them from environmental stress, including antibiotics and the host immune response. Now we're going to talk about microbial nutrition and growth. So the nutritional requirements generally include, which is the very basic, glucose, amino acid, vitamins, mineral, environmental condition, oxygen, osmotic condition, able to grow in what we've been using in lab, TSA, MSA, phenol red. Phenol red usually contains a lot of rich nutrients such as sugars. Again, like last time, facultative anaerobe can grow in the presence and absence of oxygen. The interaction that this bacteria has includes competitive interactions, meaning they just fight over nutrients. It also includes synergistic interactions, basically state that they are able to coexist with other bacteria, which is commonly found in biofilm formation. Like I said on the previous slides, the biofilm formation provides protection from environmental stress and antibiotics. They can also interact with other microorganisms from pathogen to pathogen, which enhances their collective vigilance. The environmental interaction this bacteria consists of, they can live in solid and water type environments where they contribute to microbial community. It's usually influenced by humans or animal source. Agricultural impact, livestock can also be affected because they can also be caused by infection, and produce and crops as well can also be affected. The symbiotic, as you can see by the illustration on the right side of the PowerPoint, that it exists typically on the skin. It's a very most common place. Typically, it can be treated by antibiotics. But there are some rare cases, such an example as staph infection, can turn deadly if the bacteria invades deeper into the body or enter the bloodstream, resulting in fever, joints, and muscle pain. And that is it. Thank you.
Hello everyone. Today I will talk about Staphylococcus bacteria. Staphylococcus is a type of a bacteria that exists all around us, often too small to see with the naked eye. Despite its size, Staphylococcus plays a significant role in various environments, including the human body. Under a microscope, Staphylococcus appear as a teeny spherical cells resembling berries. These cells are encased in a protective cell wall and contain essential machinery for sustenance within their cytoplasm. Staph belong to a diverse family of bacteria within over 30 identified species. One notable member is a Staphylococcus aureus, known for causing infection in humans Understanding its place in the microbial family tree help us appreciate its complexity. Staphylococcus obtains energy through various metabolic processes, enabling its growth and proliferation. Its ability to thrive in a different environment is closely linked to its metabolic flexibility. Staphylococcus relies on a range of nutrients for growth, adapting to different conditions to ensure its survival. Understanding its nutritional requirements shed light on its behavior and various settings. Staphylococcus interacts with a diverse array of microorganisms, both beneficial and harmful. These interactions shape microbial communities and communities and influence uh, ecological dynamics. Beyond microorganisms, Staphylococcus interacts with other organisms and the environment, impacting ecosystem and human health. These interactions range from symbiotic relationship to pathogenic infections. Staph has a significant impact on human health, causing a range of infection from minor skin elements to life-threatening condition. Understanding these impacts is crucial for effective management and treatment. By exploring this aspect of Staphylococcus, we gain a valuable insight into its significance in microbial ecology, human health, and beyond. Staphylococcus bacteria showcase a surprising range of interaction with the human body, while Staphylococcus aureus is notorious for causing infection. The staphylococcal family is a diverse group. Some members, like Staphylococcus epididymis, live peacefully on our skin, even con- contributing to our well-being. Grasping this spectrum of relationship, from helpful to harmful, is essential for maintaining a healthy balance. Taking a closer look at Staphylococcus reveals both the interplay between microbe and human health, as well as valuable knowledge for controlling infection. Thank you for listening.
Hi, hi, my name is Dawson. Today I'll be presenting my least favorite microorganism, Treponema pallidum. In overview, I'll be talking about history, background information, the microbiology behind it, transmission, diagnosis, and treatment. History and background. Commonly known as syphilis, this bacterium is the reason why there are over 200,000 cases of this STD yearly. The first recorded outbreak of this disease dates back to the French invasion in 1494 in Naples, Italy. In 1905, Schouten and Hoffman discovered the etiologic agent of syphilis, where they named it Spirochaeta pallida. A cure was founded for this disease in 1943, giving 4 patients penicillin. T. pallidum is a bacteria that causes syphilis. Microbiology behind it. Treponema pallidum is a gram-negative bacteria, but is not similar to your average gram-negative bacteria. T. pallidum is from the spirochete family. They are helically coiled corkscrew-shaped cells. They can be up to 6 to 15 micrometers long and 1 to 2 micrometers wide, with flagella surrounding it. The flagella gives the bacteria its shape and mobility. The spiral shape gives the bacteria the ability to spin and penetrate skin. It is anaerobic, meaning that it can grow without oxygen. It cannot be grown as a pure culture due to not meeting Koch's postulates. T. pallidum DNA was detected with the use of primers to the gene coding protein with a molecular weight of 47 kD and T. pallidum RNA with the primers to the gene of 16S RNA. It is difficult to culture because it does not use your usual gram staining technique to identify the bacteria and classification. Techniques such as the silver impregnation stain or the reuse stain will have a better result when observing the bacteria's cells. Since T. pallidum usually affects the living host, they are usually interacting with the human platelets. Transmission. There are 4 ways to contract this disease. Number 1, sexual contact in any way. 2, contaminated needles. 3, a baby can catch the infection in the womb. 4, direct contact with a skin lesion. Diagnosis and treatment. There are 3 stages when contracting syphilis. The first stage is usually painless. There's usually chances of you getting sores or chancres around your genital areas or where there was contact with the bacterium. The second stage is a body rash. It's not painful, but it's severely irritating. And the third stage can be fatal. It can occur 10 to 30 years after being in contact with the disease. It affects organs such as the brain, the eyes, the heart. There are serological tests that people can take to see if they have the disease, and there are antibiotics and a cure to syphilis, but it is best to get treatment right away if you do find out you have syphilis. In summary, I talked about the history and background information, the microbiology behind it, transmission, and diagnosis and treatment.
For my term project, I will be introducing Escherichia coli. E. coli is normally found in the lower intestine of animals and humans. It can also be found in water, soil, and vegetation, resulting in fecal contamination. Most E. coli strains are harmless and are vital components of the healthy gut flora, aiding in vitamin synthesis and digestion. E. coli does have the potential to induce harmful diseases. E. coli is a rod-shaped bacterium, also known as a bacillus. E. coli usually appears by itself or in pairs, sometimes found in short chains. E. coli is a bacteria belonging to the phylum Proteobacteria, class of Gamma Proteobacteria, order of Enterobacteriales. It also belongs to the genus Escherichia and species of Escherichia coli. Relatives include Salmonella and Shigella, along with Proteus, Enterobacter, and Klebsiella. E. coli is facultative and aerobic, meaning it can grow with or without the presence of oxygen through anaerobic respiration, or fermentation. It can synthesize everything inside their cells from precursor metabolites alone. E. coli reproduces by binary fission. Its nutrition and growth depend on various factors including nutrients availability, temperature, pH, and oxygen levels. Optimal growth for E. coli occurs around 37 degrees Celsius, while the pH ranges from 7.0 to 7.5. The types of interactions E. coli has with other microorganisms are competitive, mutualistic, biofilm formation, pathogenic, and probiotic. Additional interactions E. coli has with other organisms and the environment are foodborne transmission, mechanical vector, and commensal relationships. Mechanical vector results in E. coli diarrhea. Some pathogenic strains of E. coli include enteropathogenic E. coli, enterotoxigenic E. coli, enterohemorrhagic E. coli. Enteropathogenic E. coli causes diarrhea primarily in infants. Enterotoxigenic E. coli causes travelers and infants diarrhea in developing countries. And enterohemorrhagic E. coli causes severe foodborne illnesses.
This is my term project. It is on Bifidobacterium, and I'm just going to be using pictures from my graphic novel I made to explain what Bifidobacterium is. So in this drawing here, I'm using Bifidobacterium to represent basically a group of warriors, a part of a kingdom that fight, protect, and gain resources for the kingdom, which would be the human body, the kingdom. And Bifidobacterium makes up 90% of gut microbiota in infants in the early stages of life, and slowly dwindles down as you grow older, of course. And the most prevalent Bifidobacteria in infants were B. longum and B. breve, and they are, as I said, the most abundant in the whole gut microbiota. So the role of Bifidobacterium is to ferment lactose and oligosaccharides, which are complex carbohydrates. They basically metabolize HMOs, which are non-digestible in the human system, but because Bifidobacterium exists, it metabolizes those, and it will help boost immune system. It will help digestion and prevent pathogens from attacking. So let's go to the next picture. Bifidobacterium, let's talk about the shape and anatomy of it. It's a gram-positive bacteria. It is basically a rod-shaped, branched looking bacteria. It's an obligate anaerobe, a lactic acid producer. It's also uses non-digestible plant polymers for energy, and it has irregular borders and has a white convex. It does not produce any spores, has no capsules, and is non-motile. It also comes from the Actinobacteria phylum, which I mentioned in the third picture there, or the first picture on top. And it is very good at growing in environments from 36 degrees Celsius to 38 Celsius, which is perfect in your stomach. Along with that comes lactobacillus and bacteroids, which are prevalent in the early stages of life as well. They also help ferment lactose. As you can see, they're buddies next to each other. And essentially, what Bifidobacterium does in the environment, like I said earlier, it surrounds the intestinal tract with itself and prevents pathogens from attaching to the intestinal tract. It also not only produces lactic acid, but produces acetic acid as well, which reduces things like yeast and molds in the gut. The bacteria itself is very non-pathogenic. There's maybe one strain out there that can possibly be pathogenic, but it's very, very minimal. So as we grow older, though, Bifidobacterium naturally decreases because just as we get older, it just does. And so, here I have a picture of the kingdom itself rapidly declining, representing the human body, decaying over time. That's just how life works. And right here, although they dwindle in population, they still do their job or try to do their job, but because as we grow older over time, enterobacteria and other pathogenic bacteria will enter the body, onto the gut microbiota, and have more opportunity to attack, basically. And in the end, although they're, low in population, they still try to do their job and do their best at what they do, which is protecting the body and trying to ferment lactose.
Clostridium difficile, aka C. diff, is the main culprit of a healthcare-associated infection that causes severe diarrhea. C. diff was discovered by Louis Pasteur in the 1860s and was first isolated from the intestinal flora of newborns in 1935. It was originally named Bacillus difficile because it was difficult to culture and study. C. diff is a gram-positive, obligate anaerobe, bacillus-shaped bacterium and has a vegetative form and spore form. Because it's an obligate anaerobe, it cannot survive in the environment for very long when it's in its vegetative state. When it's in its spore form, it is metabolically inactive and can withstand oxidative stress, extreme temperatures, desiccation, and acidic environments. The C. diff spores in the stool of those who are infected can live in the environment for extended periods of time, as they can survive harsh conditions, including the surfaces that are sanitized with alcohol. C. diff is part of the phylum Firmicutes and has many relatives, including Clostridium perfringens, which can cause food poisoning and diarrhea, and Clostridium tetani, which can be found in soil and cause spastic paralysis. When C. diff enters its host, it navigates through the acidic environment of the stomach and then travels to the small intestine where it germinates. For germination to occur, germinants and the proper nutrients must be present in the environment. Bile acids, along with glycine and other amino acids, trigger the germination of C. diff spores. Glycine and the other amino acids act as co-germinants. The spores then move into the anaerobic environment of the large intestine where they multiply and colonize. They then attach to the epithelium of the colon and secrete toxins that cause the infection. C. diff is found in soil, feces, sewage, and food and is transmitted through the fecal-oral route. CCPA is the main carbon catabolite regulator that also reduces the toxin gene expression.
It uses the Stickland reaction, which oxidizes and reduces glycine and proline, which produce ATP and NAD+. When there's a limited supply of proline or proline reductase, alternative pathways are used to regenerate NAD+, which produces butyrate, which then stimulates the synthesis of the toxins of C. diff. Antibiotic use decreases the overall diversity of the intestinal microbiome. Specifically, broad-spectrum antibiotics decrease the amount of bacteroidetes, which allows germination and growth of pathogenic microorganisms such as C. diff. When the gut microbiota changes, the number of metabolites in the intestine are also affected, which alters bacterial fermentation. This decreases the amount of fatty acids while increasing carbohydrates and amino acids, which allows C. diff to use these metabolites for germination, colonization, and growth. Those who are at high risk for contracting C. diff infection include the elderly with multiple comorbidities who frequently undergo urgent surgery. Those who become infected with C. diff tend to be readmitted to the hospital, which provides the pathogen with more opportunities to spread to its next victims.
Salmonella is a pathogenic bacterium responsible for significant public health issues, causing about 93.8 million cases of non-typhoidal Salmonella gastroenteritis and 155,000 deaths annually. It originates from the intestines of various animals, making them natural reservoirs. Transmission occurs through cross-contamination during food preparation, direct contact with infected animals or their environments, highlighting the need for strict hygiene and food safety practices. Salmonella is a gram-negative, flagellated, facultatively anaerobic bacillus characterized by OH and VI antigens. Its rigid cell wall made of peptidoglycan provides structural support, while the outer membranes, lipopolysaccharides, are crucial for pathogenicity. Peritrichous flagella, distributed across the cell surface, enable movement through the host's intestinal tract and invasion of host cells. Additionally, pili help the bacteria adhere to host cells, enhancing infection. This complex anatomy allows Salmonella to thrive in various environments, reinforcing its role as a major pathogen. Salmonella, a group of gram-negative gamma-proteo bacteria, consists mainly of two species, Salmonella enterica and Salmonella bungoi. The phylogeny or evolutionary relationships of Salmonella has been extensively studied. Research, including the analysis of housekeeping genes like GAPA, PHOP, MDH, and RECA, reveals distinct lineages and subspecies within Salmonella. These genes help characterize the genetic relationships among various isolates. The study molecular phylogeny of Salmonella presents a robust phylogeny that clearly defines the lineages of diphasic and monophasic subspecies. However, the mobility of genes like RECA among different Salmonella types adds complexity to understanding their evolution over time. Salmonella exhibits a flexible metabolism, enabling it to thrive in diverse environments by utilizing various nutrients like carbohydrates, amino acids, and fatty acids. As facultative anaerobes, they can switch between aerobic respiration and fermentation depending on the presence of oxygen. This metabolic versatility and allows Salmonella to survive and grow in different hosts, enhancing its pathogenicity and ability to cause gastrointestinal infection in humans and animals. According to the text, Salmonella enterica remodels the host cell endosomal system for efficient intravascular nutrition. Salmonella enterica adapts within host cells by forming the SCV-SIF continuum, which helps it access nutrients from the host cell's endosomal system, overcoming nutritional limitations. Salmonella interacts with other microorganisms within complex microbial communities, influencing its infection impact. It competes for nutrients, resources, and space, shaping its population dynamics and ability to thrive. Salmonella benefits from the metabolic byproducts of some microorganisms, while sometimes inhibiting others' growth. Its capacity to form biofilms further complicates these interactions, affecting stability and resistance to antimicrobials. For example, Salmonella's persistence in soil relies on interactions with indigenous microorganisms, including fungi and protozoa, which influence its survival and transmission. In agricultural soils, competition with native microbiota is crucial, as soil diversity helps prevent Salmonella colonization, reducing foodborne outbreak risk. Salmonella interacts with a variety of organisms and environmental factors, particularly in agricultural settings where it affects plants, animals, soil, and water. Environmental conditions like temperature, humidity, and pH can influence its survival and transmission, leading to disease outbreaks. Salmonella's interactions range from symbiotic relationships to significant public health impacts, causing enteric salmonellosis and resulting in substantial morbidity and mortality globally. Its presence in agriculture affects food safety and soil microbiota, underscoring the need for hygiene, public health interventions, and environmental management. Comprehensive strategies to mitigate salmonella risk are essential for protecting both human health and environmental well-being. Understanding its pathogenic mechanisms and adaptability is crucial for developing effective interventions. The human microbiota is all of the microorganisms that are normally present on or in the human body. So starting at birth, the human is exposed to more and more microorganisms in its environment that eventually adapt to survive in certain areas of the body. Bacteria in the human microbiota participate in digestion, immune system regulation, protection from disease-causing bacteria, production of important vitamins, and more. The human microbiota can influence someone's susceptibility to infectious disease, contribute to chronic illnesses, or even determine how a body responds to pharmaceutical treatments. When microorganisms that belong to one bodily system or anatomical site gain access to a different part of the body, this can lead to those once harmless, or even helpful microorganisms causing disease. Increasing incidences of several atopic and autoimmune diseases over the last few decades have led to something called the hygiene hypothesis, which states that practices including the use of refrigeration, pasteurization, water treatment, and food processing implementation on a broad scale can lead to populations of children not being exposed to enough microbes. Several studies suggest a correlation between disrupted microbial composition and diseases like allergies, asthma, atopic eczema, and inflammatory bowel disease. Widespread use of antibiotics may also be contributing to the disruption of microbiota. Several studies have shown that antibiotic treatments can lead to decreases in beneficial bacteria, as well as increases in potential pathogens. Some bacterial shifts that result from antibiotics have been found to persist for up to 2 years. The mouth is a cozy place for some microorganisms because of its availability of water and nutrients and the neutral pH and its warm temperature. Some microorganisms in the mouth can resist the mechanical removal from chewing and swallowing by adhering to surfaces such as gums and teeth. Others get swallowed or shed by epithelial cells.
Bacteria and protozoa and fungi and viruses and archaea are all found in the normal microbiota of the mouth. There are almost 1,500 total genomes in the oral cavity, including 700 identified species of prokaryotes, both gram-negative and gram-positive bacteria. The microbiota in the mouth is very dynamic. It's impacted by the frequency at which food is consumed, changes in the pH, interactions among bacteria, and in the long term, gene mutations and horizontal gene transfer between bacteria. Many microorganisms in the mouth have symbiotic relationships with their host. Much of the oral microbiota exists in the form of a biofilm that helps maintain oral homeostasis, protect the oral cavity, and prevent disease development. Imbalances within the oral microbiome can lead to tooth decay and cavities. When the bacteria are able to colonize on the surface of the tooth, they may produce acids that can demineralize the tooth's enamel or cementum covering the root and the underlying dentin. With enough deterioration, bacteria gain access to the pulp chamber, and if the pulpal tissue becomes infected, this infection can destroy the pulpal tissue and extend out to the roots of the tooth and into the surrounding periodontal tissues. Streptococcus colonizes the oral cavity within hours of a human's birth. Later, as teeth grow in age, Streptococcus parasanguis and Streptococcus mutans colonize the enamel surfaces of the teeth, and Streptococcus salivarius makes itself at home in the surfaces of the cheeks and the gums and colonizes the saliva. This is aided by Streptococcus's ability to readily produce glycocalyx and other adherence factors. Streptococci are based on their hemolytic, serologic, physiological, and genetic similarities. Streptococci are divided into 6 phylogenetic groups: Anginosus, bovis, mitis, mutans, pyogenic, and salivarius. Many streptococci are members of the normal human microbiota. However, some are highly pathogenic. For example, Streptococcus pyogens can cause the infamous strep throat, and Streptococcus mutans is known to cause cavities and tooth decay. Bacteria in the Streptococcus genus are spherical or oval-shaped and are often arranged in pairs or chains. Their cells are gram-positive. Streptococci can grow anaerobically or aerobically, but cannot use oxygen for metabolic reactions. Instead, they use glucose and some other carbohydrates as sources of energy.
Hi, welcome to my favorite microorganism. In week 4, we were briefly introduced to the domain Archaea, and I was interested to learn more about these unicellular organisms, specifically how they differ to their prokaryotic friends, bacteria. And they differ in 3 major ways. One way being the lipid makeup of their cytoplasmic membrane. Bacteria tends to have straight chains of lipids, whereas the Archaea organisms will have branched lipid chains or even hydrocarbons. The next thing is they don't have a true peptidoglycan membrane, which makes them less susceptible to enzymes that will attack and break down cell walls. But due to this, irregardless of this fact, microbiologists have not found a single organism belonging to Archaea that are pathogenic. Lastly, their first initial amino acid in their DNA sequence starts with the AUG start codon, which is way more similar to eukaryotes than bacteria. Now, more specifically in the Archaea domain, I wanted to look at the phylum of Euryarchaeota, and that includes methanogens. They are the largest known group of Archaea, and they are very varied in their presentation. Microbiologists have identified 50 known species, and they can present in a cocci form, in bacilli form, and they have even been observed in a spiral formation. There's also been some that have been observed to have filamentous appendages. So very varied in their presentations. Scientists believe that methanogens exist on the edge of life. And that is because these organisms can grow autotrophically in the absence of organic substrates, absence of oxygen, and in the absence of light. This vast group of Archaea consists of thermophiles and halophiles, and they can be found in the guts of animals. They can be found in hot springs or deep sea hydrothermal vents. They are also found in oil wells and in landfills and in the sediment beds of marine plant matter. So with that, These organisms are also the only ones that can produce methane gas, and they do this anaerobically as a way to conserve ATP. They rely on access to substrates like carbon dioxide, dihydrogen, formate, acetate, methanol, methyl sulfates, etc. All those things I mentioned are waste materials from other organisms, which makes methanogens the ultimate cleanup crew of kind of toxic materials. And so they're attractive to humans as we look in ways to further degrade and break down our waste materials. Another thing that I wanted to look at as far as methanogens is their role in our current global crisis of global warming. And the warming potential of atmospheric methane has a 30 times greater warming potential than carbon dioxide, and that is over a 100-year period. And one of the biggest contributors to atmospheric methane, is the agricultural industry. And that is completely a man-made problem. But there are more studies happening with how to change the diets of these animals. One cow and the microbes that live in its gut has the potential to produce 150 to 500 grams of methane a day. So lots of studying going into adding materials to their feed or changing their feed, which would reduce the access to the substrates mentioned above for the methagenesis production. Other sources like landfills and sewage treatment centers are figuring out ways to capture that methane before it reaches the atmosphere and turn it into power. So my art project was actually just further expanded on someone else's art. So this represents the sewage water waste facility in San Jose, California. They are very tall, 70-foot tall towers. And the artist, Buster Simpson, put methanogen organisms in their microscopy pictures on these towers. And they are backlit by LED lights. And it just is illuminating the fact that this whole building is powered by the methane that these microbes release. And so I took it a little bit further and just put, power lines to see how we can utilize that in our homes and in our towns. So we can warm our homes with the methane versus, warming the earth. That is my presentation. Thank you.
Imagine all the world's greenery returned to violet, to a time when our ancestors had not yet dreamed of chlorophyll, and when all the sun's light was harnessed by retinol instead. Science fiction or fact? The answer lies in the extremes of life. For my term project, I have chosen Halobacterium. a genus from domain Archaea whose closest relative is the methanogens. Halobacterium is of particular interest to astrobiologists who are searching to answer one of life's biggest questions: are we alone in the universe? Astrobiologists are particularly fond of Halobacterium because species members are polyextremophiles. This means that they are adapted to survive in multiple extremes under multiple stressors like those that may be encountered in planets outside our own, like the surface of Mars, for example. One of the most fascinating features of Halobacterium is that they produce spectacular blooms that are visible from outer space. And some scientists believe that when oxygen levels on Earth were low about 4 billion years ago, Halobacterium may have painted the Earth's surface purple long before chlorophyll painted it green, and you may have noticed it from outer space. As the prefix on the name implies, Halobacterium thrive in hypersaline environments. However, the suffix bacterium in this case is a misnomer, since scientists now know that this microorganism is not a bacteria but a member of Archaea instead. They share many common features with other members of Archaea. They are pigmented, they are bordered by a lipid monolayer containing ether-linked phospholipids enclosed within a glycoprotein cell wall. They are flagellated, which allows them to swim to places where growth conditions are optimal. They are generally rod-shaped, but some species members are highly pleomorphic and can exhibit some unusual shapes. And rather unique to them is the presence of glass vesicles that allow them to float. Salt is of course required for growth. They are easy to culture in a lab setting as they can be grown from liquid culture, colony to liquid culture, and even cell colonies. They are metabolically flexible. That is, they can grow both in aerobic and anaerobic conditions. Under aerobic conditions, they can obtain energy through cellular respiration by oxidizing various metabolites. And when oxygen levels are low, they can generate energy through fermentation of the amino acid arginine. But their most unique characteristic is that they are the only member of Archaea that can generate energy via photosynthesis. And unlike plants and animals that use chlorophyll for photosynthesis, they possess their own unique protein complex. Bacteriorhodopsin is the sole protein found on the purple-colored patches of the purple membrane of Halobacterium. The purple color is caused by retinol, a molecule that absorbs light at a particular wavelength and emits color as a result. In the case of Bacteriorhodopsin, the retinol is bound to an integral peptide protein that absorbs mostly green light and transmits purple light. This is why it appears purple. The discovery of Bacteriorhodopsin is kind of a big deal because up until that point, it was believed that these light-sensitive pigments only existed in animal eyes. So how does it work? When the retinol absorbs a photon of light, the shape of the molecule changes, and this change causes a conformational rearrangement in the surrounding protein. This allows for Bacteriorhodopsin to behave as a proton pump, moving hydrogen ions against their electrochemical gradient from the cytoplasm of the cell to the extracellular space. So for each proton absorbed, one hydrogen ion is taken out of the cell. The accumulation of hydrogen ions in the extracellular space activates ATP synthase as hydrogen ions move down their electrical chemical gradient back into the cytoplasm of the cell. Now, communities of hypersaline environments tend to have low complexity because competition is significantly reduced in such extreme environments where only those adapted to such harsh conditions survive. But Halobacterium is not the only organism that's adapted to survive here. The extremely halophilic red-orange bacterium Selenobacter compete for space with Halobacterium in these hypersaline environments. So does Dunaliella salina, the unicellular green algae that turns orange under abiotic stress. And most surprising, the haloviruses which actually infect Halobacterium. Now, if you remember, Archaea is not known to cause any diseases in any humans, animals, or plants, but this doesn't mean that they're immune to viruses. Halobacterium raises environmental red flags. When a railroad causeway was built in Utah's Great Salt Lake in the 1950s, it divided the lake into a north and south arm, cutting the north arm from the influx of fresh water and raising its salt content to double that of the south arm. In the north arm, Dunaliella salina and Halobacterium thrive. Despite the beautiful color displays, this was not good for the other organisms that previously existed in this ecosystem. Brine shrimp, brine flies, green algae, and all the migratory birds were no longer there. All the living communities had been reduced to the microscopic level, causing a bit of a man-made environmental disaster. When a pond in Maui turned bright pink, it serves as a warning sign that increasing temperatures and drier conditions made the water saltier, allowing for halobacterium to thrive and for everything else to pretty much die. So the presence of halobacterium could spell trouble for animals that rely on these important marine environments to survive. Scientists warn that our future may hold more pink lakes, that is, more halobacterium, as the planet warms.